Okay, let's get started. Today then we're going to finish off very quickly talking about signaling, the action potential, generated potentials, and then we'll start very briefly touch on fiber types, and then we'll start getting into the cord itself, looking at the meninges, and hopefully looking at some of the pathways within the cord. Quick clarification. We were talking about neurotransmitters. Now, the effect of a neurotransmitter depends on the receptor it attaches to. We haven't gone over all that yet, but we're going to get to that later when we look at specific regions within the brain. So these are the class list on the bottom. You've got acetylcholine being excitatory or inhibitory, depending on which neurotransmitter it goes to. Glutamate, excitatory. Dopamine being inhibitory. But some of these neurotransmitters have more than one effect. So you can't always say dopamine is an inhibitory neurotransmitter. It's inhibitory within the nigrostriatal system, but in other places, depending on what receptor that tissue, that neural cell expresses, dopamine can be an excitatory neurotransmitter as well. So it's not always black or white. Unfortunately, it gets more complex than that, and I've tried to skip over that complexity. For example, GABA, which is the most common, prevalent, well-known, cited inhibitory neurotransmitter, gamma-aminobutyric acid. That, in fact, some research has shown that can be an excitatory neurotransmitter in certain parts of the nervous system, depending on the receptor that it binds to. For example, dopamine is like 7 different types of dopamine receptor. We haven't gone into all those, D1, D2, 3, 4, etc., etc. I don't think I'll go into those, but just be aware that it's not as clear-cut as some of these tables we show. Sometimes the neurotransmitter can be excitatory, even though most of the time it's inhibitory, etc., etc.
Sorry, this has nothing to do with lecture, but this machine is really loud and it's hard to hear. I don't know if it's the speaker or...
I've got no speaker on. As long as this sound system is off. I don't know what's in here. If I click any buttons randomly. No, I don't seem to have any sound system as long as this is off. I don't see any volume control. I'll just shout louder or... There we go. Okay, so back to neurotransmission, and we started to talk about the action potential, right? And just a very brief, this is my take on how you get a action potential. You've all done this before, I think, surely in your AMP, an action potential. So here is my non-mind axon. Okay? And remember, to get work, to get an action potential, before you get an action potential, you have to generate this battery kind of thing with the cathode inside and the positive outside, the anode outside. And that's usually about a 70 millivolt, 70 microvolt signal, difference, right? And that is, of course, a function largely of a sodium-potassium pump. It pumps that sodium out, whatever, okay? So, let's now get an action potential going. I stamp on this neuron, I stamp on this nerve cell. And what we call, by doing that, we change that threshold, we change that electrical potential. it goes from, as you can see on this graph, from minus 70 gradually up to, let's say, minus 55. If you hit that threshold, of this case it's minus 55, below that nothing's going to happen. You can prod that nerve and nothing happens. And it goes up to, minus 60, nothing happens. But when you hit a certain critical threshold, in this case minus 55, typically what you'll see in text, the ion channels suddenly open and you get a flood of sodium into the cell, into the axon. Okay? And you get actually a positive signal. Might go up to about plus 35. You can see on this graph, on there, I don't know that thing's even aiming on the right place. It goes above, so you actually get a positive inside and negative outside. That's called depolarization, right? Obviously, you've heard this before, right? And what happens then, that wave of depolarization goes all the way down the membrane. So that bit signals the next bit, and you get this wave of depolarization and repolarization, and that's your action potential. Simple as that. That change in electrical potential across the membrane, that wave, remember, goes all the way to the end without speeding up, without stopping, without increasing amplitude or whatever. Okay? Immediately after that depolarization, there's what's known as the absolute refractory period. You've heard of that term? Absolute refractory period. It lasts about half a millisecond, which is half of a thousandth of a second. What does that mean? What does the absolute refractory period mean, functionally? What is the absolute refractory period?
No matter the stimulus, it's not firing.
No matter how hard you stamp on that axon, it will not fire. Absolutely no chance will it fire another signal. And that lasts about for half a millisecond. But after that, between about a half a millisecond and 1 millisecond later, you get into what's known as a relative refractory period. What does that mean?
Can you repeat what the absolute?
The absolute refractory period is a period where no matter how high the stimulus is into that axon, it will not fire. It's dead. You can't make it fire. So there's a delay after that initial signal goes through it, that depolarization, repolarization. Then we get a period of time after the absolute refractory period is finished, which lasts between roughly half a millisecond to a millisecond, which is half of a thousandth of a second to 1,000th of a second, where you get what's known as the relative refractory period. What happens there? Why is it called relative? Adam?
It takes more stimulus to fire it than normal.
You can fire it during this stage, but it's not as easy to fire it as it was if you started off the resting state. So in other words, you can fire an axon, a nerve cell, during the relative refractory period, but you've got to be pretty serious about stabbing that thing. You've got to really put a lot of stimulus into it for it to fire. So this is how we get changing signals going down the axon. Signal information. Remember, a pistol. If I pull the trigger harder, I do not get a faster bullet. If I pull the trigger harder, I do not get a bigger bullet coming out of the end of the gun. But the way I can send information is like Morse code. I can fire my trigger at a faster rate. And that's how big stimuli cause increased rate of firing. If you're a weak stimulus, you fire the axon and then you have to wait past the absolute refractory period to start firing again if you're weak. But if you're a strong stimulus, you can make the axon fire earlier because you can fire within its relative refractory period, and therefore you get more firings per second. Therefore, the strength of the signal, the strength of the stimulus is coded with FM, frequency modulation. That makes sense. The more frequent the nerve cell fires means there's a stronger stimulus coming in from somewhere. It's not the amplitude change, it's not the speed change, it's the frequency. So you code information using FM in an axon. That makes sense. Okay? So that's what's known as an action potential. It goes all the way down. Okay. So, this is a non-myelinated axon. What happens when we get a myelinated axon. So here's my... And now we get some myelin here. We get these Schwann cells producing our myelin sheath like that. Okay? How is the signal different in a myelinated axon than a non-myelinated axon?
It's faster.
It's faster, yes. Fair enough. Why?
Because it skips the myelination.
It skips the myelination. It's called saltatory conduction, right? What do you mean by skips the myelination? How does it skip? So you've got this signal here in this non-myelinated piece coming down to... And then what happens? It skips, it jumps. How is that possible? The myelin, right, prevents any depolarization beneath it. There are no ion channels beneath the myelin sheath. You cannot depolarize and repolarize the membrane beneath the myelin sheath because there are no ion channels. This Schwann cell, this myelin sheath, is preventing that. Okay? So what happens then? Well, the signal dies off. The signal, so this is an active process, right? You all agree, depolarization and repolarization is an active process because you have to depolarize after it's been, you've got to repolarize after it's been depolarized. You have to use that sodium-potassium pump to get that electrical gradient back up to where it needs to be. Active process, slow, requires energy, inefficient. I want something faster than that. So what you do is you put a myelinated axon there. And what happens, so you've got this signal, this depolarization and repolarization signal coming down to myelin. It stops. It can't go any further. So what happens? It passively spreads down the axon underneath the myelin, like an electrical wire would. Just as so your cable-like properties beneath the myelin. It just goes, the electrical signal flows through the axon passively. Just like if I put an electrical current down a wire, it goes passively. There's no active process to get the electricity through these wires. It's all passive. It's been sent from a generating plant somewhere. It's pure passive. Okay? And that's exactly what happens beneath a myelinated axon, within a myelinated axon. And passive is way faster than active. Way faster. I don't have to depolarize and repolarize the membrane. It's just passive. No energy required. Passive. Saves energy. It's faster. But there's resistance within our axon. So it's going to die off. Just as there's resistance in an electrical wire. If I put a car battery against a wire here and half a mile down the road there, you held the end of the wire, you wouldn't feel it. Okay? It's because there's resistance in that copper wire. There's resistance in the axon. So that signal strength, that passive signal, basically dies off very quickly. About a millimeter or so, it's gone. Okay? But fortunately, just before it dies off, you stick a node of Ranvier there. You have a gap. And that signal strength, as it just gets that node of Ranvier, is just enough to cause the dipole, that signal we see on the board, and we get another big booster station, fires, it depolarizes the membrane in that node of Ranvier. And then it passively spreads down to the next one, and just before it dies, it gets another boost, etc., etc., etc. So you get depolarization at the nodes of Ranvier, but you get a passive spread beneath the myelin sheath. That's how it skips, as it were. That's how it jumps, as it were. It actually passively spreads beneath the axon, and that's much faster. That's why it saves energy, but you have to build Schwann cells around them, and that's why you get fast conduction. Way faster conduction in a myelinated axon than a non-myelinated axon. So a a myelin axon faster, how is it another way we can increase the speed of transmission?
Increase the thickness of the axon.
Diameter. You make a thicker axon. So this axon will transmit a lot slower than that axon. Okay? And it's just, have you seen the reason why, or the speed, is going to be lambda is square root of Rm over Ra. That is, lambda is directly proportional to the speed of conduction down an axon, right? Ra stands for axonal resistance. Rm is membrane resistance. Square root of that. That's lambda. And the fact is, your membrane resistance doesn't really change however fast or however small you are. But axonal resistance does. The resistance in a wide, fat, big axon is much less than a thin, small one. And therefore, as Ra decreases, lambda will increase and the speed will increase. It's just like a copper wire, right? You get a thin copper wire, you stick electricity down it, it gets hot. It's resistance. That's how not these lights, but your incandescent lights work, right? Those little light bulbs, they glow because you've got a very, very thin piece of wire you're chucking on a current through. And it can't handle it, so it glows. The thicker an electrical wire is, the less resistance there is to it. There's less likely to heat up as you stick current down it. A thin wire heats up because of resistance. Same thing with these axons. The bigger the axon is, the faster it will go. Okay? And here you can see some examples of speeds, conduction velocities. Okay, let's look at on the left hand side first. There are different ways of classifying axons, fiber types. Have you seen this? A, B, C. You've seen this before, yes? No. And it's based on the diameter and it's whether we're myelinated or not. So on the left hand side we see fiber types. And you've got A, B and C. A and B are going to be myelinated. And within the A we've got these categories of A alpha, beta, gamma and delta. Okay, you can see where those things occur. And look at the velocities on the far hand side, the diameters and the velocities. Velocity here of A alpha, alpha motor neurons for example. Those are the biggest ones we get. Where do we find alpha motor neurons? What do they do? Where do our alpha motor neurons go? What's their function? Muscle, right? Go from a spine to muscle. We want to contract your muscle pretty quickly. That's important. Okay? And so they're going to be one of your biggest fibers. And they're going to be myelinated. You can see A alpha on the left hand side there. Proprioceptus, skeletal muscle, whatever. Big fat axons, myelinated. They're going to go fast. And they're going to go, we've got here like 120, 70 to 120 meters per second. That's about 270 miles an hour. So those things are flying. 270 miles an hour to conduct down that. Very, very fast. I mean, that, you think well, yeah, it is fast, but I mean, like something like a copper wire will be transmitting at 600, 700,000 miles per second. That's what copper wire transmits at. So 120 meters per second is pretty fast, but nothing compared to, you know, man-made materials. And as you go down, you see the diameter decrease, and the speed consequently will decrease. B fibers, they're still myelinated, but C fibers, these are non-myelinated fibers now. Look at the speed there, half a meter per second, roughly, or to 2 meters per second. Much slower, much, much slower. And that's because they're non-myelinated, that's C fibers. Those are our pain fibers, remember? That's why sometimes you put your hand on a hot plate, and you pick your hand up and you go, ow! I feel like 3 seconds later. It's not 3 seconds later, but you feel a delay there because, you know, your pain fiber is slow transmitting compared to other types of fiber. We'll come back to fiber types in a minute, but we must remember those names, those fibers. I don't expect you to remember the speeds, but the relative thickness of those fiber types. Alpha, beta, gamma, delta, B and C. The relative order of those are important in determining fiber type, fiber size, and therefore velocity. And we'll come across these where we find them in the nervous system in a little bit perhaps. Okay, so this is what's action potential, right? How we I skipped ahead, but how do I actually signal this thing to start? How do you typically find an axon firing? What causes an axon to fire typically? You've got your cell body, haven't you? Here's your cell body with all your dendrites, and your axon hillocks here, right? That's what typically will cause an axon to fire, we hope. That's a normal course of, and what we get, remember, is we have got these synaptic, these presynaptic terminals here onto these dendrites. Okay? Is there all or nothing conduction down to the cell, to the dendrites, to the axon hillock? Is that the same as this? Depolarization membrane? No. What is it? What's it conduction called? Decremental conduction, which is a passive thing. So these are sending their signals towards the axon hillock, and they're gradually dying off, remember. But they add up spatially and temporally to get some signal to the axon hillock. And that's what's known as a generator potential versus this action potential. You need a generator potential to get an action potential. They generate the action potential. Okay? So if you get enough signal strength at the axon hillock, you'll fire the neuron. And do you have enough generator potential? Do you get that depolarization to, minus 55 to be able to fire that neuron at the axon hillock? So this information, this generator potentials are coded how? This is coded frequency modulation. This is coded how? Amplitude, AM. This is the AM radio. AM and FM. This is amplitude modulation. In other words, the more amplitude, the stronger the signal. You don't increase amplitude in the action potential. It's the same thing all the way along. Okay, does that make sense? So you've got to get enough generated potential at the axon hillock to cause it to fire, and then you fire it. Simple as that. And then you get this all or none all the way down the end. Either slow action potential if it's a non-myelinated axon, or a fast one if you've got a myelinated axon. Okay? So, again, fast fibers are your A fibers. Slow and B, slowest are your C fibers. Your A fibers are the biggest, but they're more, because of that, they're more prone to injury from ischemia and other kind of pressure palsies, nerve palsies, kind of pressure restriction, and you get a brachial plexus nerve palsy. These are the neurons you're going to find damaged first. Okay? Your non-myelinated fibers, like your C fibers, the small diameter fibers though, are more easily stimulated artificially by neurons. When you're using E-stim, if you ramp up, you've done E-stim, yes? Over Christmas, whatever it was. As you gradually ramp up the electrical stimulation you're putting into somebody, you're going to hit the small fibers first. The C fibers are going to be activated earlier. If you ramp it up, then you get the medium, and then ramp it up, you get the motor at the end. Motor fibers are much harder to stimulate, right? If you've done that in your E-stim, I mean PTs anyway. Those are harder. You have to really zap a person, don't you? And it hurts after a while. Russian stim, you've already, some of you experienced Russian stim, when your leg is out of, you can't control it. It hurts like crazy after a while. It's not very nice, is it? That's because you're pumping a lot of current in the person to get to those big motor fibers. You can hit your C fibers with less E-stim. Your small fibers, of course, small diameter fibers are also more easily affected by anesthetics and things like this, fortunately. It's the C fibers, your drugs you stick into somebody, you're going to kill those, or kill them, but inhibit those fibers before you inhibit your motor fibers. Okay. Same with ice. If you can actually deafferent somebody. You can prevent sensory information going into the cord by giving an ice cuff. You put an ice cuff around somebody. You're going to inhibit the small sensory fibers before you hit the motor fibers. So you can actually deafferent somebody temporarily by using ice for experiments. They've got the motor fibers still working, but the sensory have gone because of this ice issue. Okay? So we've got, unfortunately, we have two ways of classifying neural fibers. We have this Greek alphabet. A, alpha, beta, blah, 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 right? But we also have this classification on the left-hand side here, the Roman numeral ones, and these are found only for sensory fibers. So you've got two ways of classifying fibers, unfortunately. Don't blame me. We need to know both. We need to know what corresponds to what, because in texts and in exams, they will give you one or the other. Yeah? They're not going to always use one or always use the other. They're used interchangeably. So 1A, usually called 1A, 1B. So 1A fibers, for example, are fibers coming from a muscle spindle, are afferents from a muscle spindle. Okay? And those are larger. They correspond to the A alpha type. Big ones. Fast. Does anybody know what 1A coming from muscle spindle signals? Remember, as you can see here, we've got 1A coming from muscle spindle. We also have type 2 coming from muscle spindle, the flower spray ending. So there's two endings within a muscle spindle. Are you aware of that or not? You've done the muscle spindle in A&P, phys, exercise phys, whatever, yes, no? No? What does a muscle spindle sense? Stretch, right? Stretch. Fair enough. The 1A fiber coming from a muscle spindle senses the velocity of stretch, the rate of stretch. We're going to come back to this. We're going to go a whole lecture on the muscle spindle. It's a very interesting, very complicated thingamajig, which is very important for therapy. Very important in rehab to understand what the muscle spindle does. A lot of our treatments like PNF patterns, hold, relax, all this stuff is based on these thingamajigs. The type 2 is a smaller fiber, called A beta. It comes from muscle spindle. That senses the amount of stretch, the static length. So type 2 will give me, I have been stretched to 5 inches, whatever. 1A says, I'm being stretched at 20 meters per second, whatever, 5 degrees per second. So that gives you the rate. Type 2 gives you the amount. And they're both important in control of our movement. And we'll get to that when we talk about muscle spindle. 1B, for example, comes, there's just examples here. 1B GTO. What does the GTO sense? Force, yeah, the force going through the musculotendinous junction. Then we've got type 3, pain, temperature, smaller, corresponds to A delta, and then type 4 is another pain receptor, corresponds to C. So unfortunately, there are two different classifications of fiber type, and we need to know both of them, how they correspond to each other. So look at those two graphs, those two figures, and basically memorize not the speed, but the relative size and example of what these fibers do. I'm going to come back to those during the whole semester. Okay. That concludes my very, very, very quick, brief overview of microscopic neuroanatomy. Piece of cake, right? Nothing new there, hopefully. Let's start looking at macroscopically now. We're going to look at the cord, go into the cord, see what's within the cord, and I'll work our way up to the brain gradually. So we're going to go macroscopically from the cord upwards. Okay, the cord starts where? These things need to be adjusted down, don't they? What's the top? Where does the cord start at the top? What structure in the body do you say, that's where the cord starts? What's that big hole in the bottom of your head called? Foramen magnum, right? Which is in what bone? Occipital bone. That's where it starts. Below that's the cord. Above that is what? What's the next thing immediately above the cord? come on, this is common sense, right? This is A and P, just pretending you don't know. You know it? What is it? Somebody tell me. Medulla oblongata, medulla, the lowest part of the brainstem, right? You hit the brainstem, and the lowest part of that is medulla, alright? So you've got the cord starting at the foramen magnum, going down and stopping around about where? Where's the cord stop? L2, something like that, L2 roughly. Okay? And at the bottom, at that point, L2, you get the conus medullaris. This cone shape swelling at the end. Right about L2. Basically, the spine grows faster than the cord. Okay, so the cord does not go all the way down the vertebral frame. It stops around about L2-ish somewhere. The nerve roots, so I've got to get back down to the intervertebral foramen to come out at L5, etc., etc., and that's how you get your chorda equina, right? Your horse's tail, the chorda equina. Those nerve roots starting at the conus medullaris, going down in the backbone and coming out at various places. You've seen that, hopefully most of you did, I think, in anatomy, right? We did a laminectomy, when we cut the jawbone, and there were those spinal nerve roots down in the horse's tail. Okay? How many pairs of spinal nerves do we have? How many pairs of cranial nerves do we have first? A dozen, right? A dozen? Then we get our spinal nerves coming out from all segmental levels all the way down the spine. How many do we have? 31. 8, 12, 5, 5, 1. Remember that? We did that anatomy. 8, 12, 5, 5, 1. 8 cervical, 12 thoracic, etc., etc. 8, 12, 5, 5, 1. How many bones in the spine? How many vertebrae have we got? Including the sacrum and the coccyx. How many got? 30. Any advance on 30? 33. 7, 12, 5, 5, 4. So you've got 7 cervical, 12, etc. 7, 12, 5, 5, 4 bones. 8, 12, 5, 5, 1 nerves. Okay? We have 8 cervical spinal nerves, but only 7 bones. Okay? Because C1 nerve root comes out above C1 bone, right? We've done this, right? You know this. Okay? And it carries on that way, so C2 comes out beneath C1 and C2, etc., etc. C8 comes out below C7. T1 comes out below T1 as it were. So T1 nerve root is going to come out of the intervertebral foramen between T1 and T2. T2 nerve root comes out between T2 and T3, etc., etc., all the way down the bottom. Okay? So those are spinal nerve roots. The meninges, however, do not stop at T2. L2, should I say. The nerve root, the meninges do not stop at L2. They get right down to L5 or even lower. What are the meninges? And why are they important? Clinically. Why is it important clinically to have the meninges to go lower down to L5 but not the cord itself? Who cares? Well, if you have a spinal tap, you would care. Okay? Or an epidural. Why? What's a spinal tap? Besides the rock and roll band.
Taking fluids from your spine.
What's that?
Taking fluids from your spine.
Taking what fluid from the spine? Cerebrospinal fluid. Not drawing blood out, obviously, right? You're drawing cerebrospinal fluid out from where exactly? Where is your cerebrospinal fluid found? Between the pia major and arachnoid, right? In the subarachnoid space. We'll talk about this now and then. Okay? So, what you do when you get a spinal tap is you bend over, grab your knees, as it were, lie sideways, and stick a dirty great needle in your back, right? and they stick it between you, have you crouch over, bend over so it opens those vertebrae out, and you stick a big needle in, and you want to go above L2 or below L2? Below. You may, as a doctor, a bit jittery, he had too many drinks the night before, and he goes above, L1, and he goes a bit too far, he hits your cord. If you go at between L4 and L5, or L3 and L4, usually one of those places they'll go in there, you will not hit the cord with your needle, will you? Because there's no needle there. It's your cord Aquinas there. Okay? But the meninges are there. So you can sample cerebrospinal fluid there and look at various things in it, talk about later, without potentially hitting your spinal cord. That's clinically why you want to, that's a useful thing for us to have. The cord finishing higher than the meninges. Right, speaking of meninges, let's look at a quick overview of a meninges, and then we'll look at the cord itself. I presume in AMP you have gone over the meninges and what they do, etc., etc. Yes, people are nodding, so we'll go fairly quickly through this then and get to the cord itself. 3 meninges, right? 3 layers of protective tissue around the cord and the brain itself. Exactly the same names as they go around it, but the whole central nervous system is enveloped in these 3 layers. Okay? And the outer layer is our dura mater, remember. Our dura mater. Dura is as in durable, mater, mother, so it's the tough mother. Tough mother, there's nothing else after mother, just tough mother, you stop there, okay? So the tough mother is this big, this yellow thing here. This goes all around the cord, very tough connective tissue. When you do a laminectomy in anatomy, you cut it, it looks like plastic, thick, leathery plastic, kind of very strong material. When you do your brain dissection, it's very hard to get the brain off sometimes. That really sticks to the bone, it sticks to the brain. It's very, very hard. Above or outside of, between the spine itself and the dura mater, we have this fatty layer, fatty space. This is the epidural space. The epidural space is a cushioning layer between the dura mater and the bones themselves of the spine. So your epidural space, then you get the dura mater. Okay? Fat and blood vessels within that epidural space. Beneath that, the next layer down is this arachnoid layer. Okay? And theoretically, there's a little space, this is really a potential space. There's no real gap between the dura mater and the arachnoid layer. It's known as a subdural space, but it's really not much there. They're touching each other. Hopefully there's no space typically in that. There's no gap in that subdural space. So then you get the arachnoid layer. It's a thinner layer. Why is it called arachnoid?
Spider web.
Spider web, because it sends projections proximally or inwards, looks like a spider's web. So you can see them here. These little yellow lines here represent those spidery kind of projections. Because they send those projections inward to connect with the next layer, which is the pia mater, the light blue on this one. Pia mater means gentle mother. Very, very thin, membranous sheet. The gentle mother. This folds around every little crack, nook and cranny of the brain and the spinal cord. It sinks into every single crack within the brain. Very thin, very gentle, very small, thin layer, the pia mater. So the arachnoid, so beneath the arachnoid, between the arachnoid layer and the pia mater is the subarachnoid space. And that's represented in this by that black, not black, but dark brown thing here. The cursor's not showing up on there for some reason. So, this piece of rubbish. Whatever. So between the pia mater and the arachnoid layer is the subarachnoid space held open by these spider-like spider web projections, and that is where we find our cerebrospinal fluid. That the subarachnoid space that contains that cerebrospinal fluid. Okay? The whole of these systems are held together, or these three sheaths are stabilized within the spinal cord, within the backbone, by these things here. The denticulate ligaments. You can see them going from the side like this across, and you can see them here as well. The denticulate ligaments. They stabilize. No, no. They stabilize these three layers and hold the sort of cord so it doesn't bang around too much within the backbone. Whatever. You see denticulate ligaments there. These are directly taken out of Nephros Atlas. So you've got Nephros Atlas on anatomy. These pictures are all in there. You can see them. Okay. So those are the three layers of meninges and meningeals that go all around the cord. Those three layers also go all around the brain. Okay? So the cerebrospinal fluid circulating within the subarachnoid space surrounding the whole of the central nervous system. The major job of this is a shock absorber, probably. Hydraulic pressure, basically the whole nervous system, central nervous system, floats in cerebrospinal fluid. Dampens, bangs or whatever. As you move around, it's a dampening kind of system. Right? If we go up to the brain, though, there's slight change. You can see on this bottom diagram here, you can see the cord ending, the meninges going lower. That's where we get a spinal tap from. But if we go up to the top of the brain, the dura mater separates out in certain places. Am I going too slow? Is this all common sense? Should I no? He's not. I mean, if you had this in AMP and you've had it 5 times before, I can skip something more useful. No? Okay. So you see on the right-hand diagram here. I'll get a better one. This is a better one. Your dura mater, in this case, it's the green line. There's my dura mater. And what happens in certain parts of the brain, that splits into two. Okay? And you have the distal, or the outer layer, sticks right along the bone of the skull. Okay? And that's called the periosteal layer. Or endosteal layer. Same thing. The periosteal around the bone, or endosteal within the bone kind of thing. So that's the endosteal or periosteal layer. So the dura mater split. The periosteal sticks with the bone, but this green line here goes inwards. And that's called the meningeal layer. So the dura mater splits into the periosteal and meningeal layers, and it splits and makes this blue space here. And that is what's known as a dural sinus. A dural sinus. A sinus, a gap within the dura mater. A dural sinus or a venous sinus. And those venous sinuses are found within cracks within the brain, within fissures. And by far the most important of these is this longitudinal fissure here. Remember, it separates the left and right hemispheres. And that, of course, is known as the superior sagittal sinus. Here. The superior sagittal sinus is the biggest of our dural sinuses, the most important. We have other ones as well. We'll talk about these in more depth when we talk about cerebral blood flow. But okay, for now all you know is the dural layer, the dura mater split, allowing pools of blood to collect within it. The dural sinuses, particularly the superior sagittal sinus, is the place most of our blood from our brain gets out. It's the start of the venous outflow of blood from the brain. Blood gets up to the brain through what two major arteries? Internal carotids and vertebral, right? And once it goes through the brain, etc., etc., does it stop? It needs to get out, and it gets out by using these cerebral veins, sometimes they're called bridging veins. These veins dump blood into the superior sagittal sinuses. Okay, that's where we collect our blood from the cerebrum. Okay, they're also important in our cerebral spinal fluid circulation. Okay, this is where we put the cerebral spinal fluid back into the blood system through these little doohickeys here, arachnoid villi. So this is a light grey, brown, pinky kind of colour here. This is my subarachnoid space. That's where my cerebrospinal fluid is circulating. And it gets back into the bloodstream by going through these arachnoid villi, which pump it back into our sagittal sinus, into our dural sinuses. Superior sagittal sinus being the most important one. Its cerebrospinal fluid looks like egg white, or thinner than that. Very like synovial fluid almost. No blood in it. Should not have any blood. If you do a spinal tap and you see blood in it, you've had an arachnoid hemorrhage, you're in a mess. Okay, they'll test it for that. Remember this, it's besides shock absorption, cerebrospinal fluid acts a bit like the lymphatic system the rest of the body. Remember, the brain does not have a lymphatic system like the rest of the body does. So the cerebrospinal fluid acts a bit like a lymphatic system getting rid of waste products and bad things. You'll find no blood in it. High concentration of proteins, high concentration of sodium, high concentration of sugars. If you find loss of sugar, if you take a spinal tap and you find this person has very low sugar within the cerebrospinal fluid, good or bad? What does that mean? What's the worst thing that can mean? Bacterial infection. Bacteria like to live on sugar. If you've got little sugar in your cerebrospinal fluid, you might have a serious bacterial infection, you might have streptococcus, you might have bacterial meningitis, whatever. That's pretty bad if you find cerebrospinal fluid without any sugar in it. Where is that cerebrospinal fluid produced? It's absorbed in the dural sinuses, right? Via arachnoid villi. How do we make this stuff? What cells, we mentioned this before, what cells produce cerebrospinal fluid? Type of a glial cell, right? Ependymal cells, remember that? Ependymal cells make cerebrospinal fluid. And they make it in our ventricles. These little red thingamajigs here line, there's clumps of these ependymal cells known as choroid plexus. And choroid plexus are groups of ependymal cells lining our ventricles. These are responsible for filtering blood. Basically, they take part of the blood and stick it into the cerebrospinal fluid, stick it into the subarachnoid space. They'll basically, cerebrospinal fluid is a filtrate of blood. So they'll take the cerebral, they make the cerebrospinal fluid from the blood, as it were, and stick it into the subarachnoid space. How many ventricles have we got? How many ventricles? 4. 4 ventricles. The biggest are by far the lateral ventricles. You have 2 lateral ventricles. these big horn-shaped things that go right through the deep within the cerebrum. Remember that very first day I showed you effacement of those. You can see here, very important to look at these in a CT scan. These look, they're not normal over here, but this is what they look like. There's our lateral ventricle, one here. It's gone on this side because of a dual hematoma. You'll see tumours, brain tumours, sticking. If the shape of these ventricles change, something's wrong. So they're kind of useful diagnostic tools to look at the shape and the location of these ventricles. If the brain pushes around and changes shape of these things, something's up. So we have two lateral ventricles. You see them here again, which then communicate with a third ventricle in midline. They drain into this third ventricle. Down in here. The third ventricle, and then finally to the fourth ventricle down here. Okay? Third ventricle here, fourth ventricle here. The third and fourth ventricle communicate via this cerebral aqueduct. A little pipe that connects the third to the fourth ventricle, called the cerebral aqueduct. Okay? So it's pulled in the lateral ventricles, pushed down because pressure builds up. It's pushed down to the third, and then pushed down to the fourth, and then pushed out down around the cord and around the brain. There's another. So the cerebral aqueduct is a potential bottleneck, a potential site of restriction. It can get clogged, as it were, to be too narrow. You can also get a restriction as you try to get out of your fourth ventricle. The fourth ventricle communicates with the cord via the median aperture. The median aperture is another little pipe, a hole, where you get out of the fourth ventricle. So you've got two potential bottlenecks here. Restriction. Okay? So it's produced in the arachnoid, by these choroid plexuses. It's pushed through the ventricles, and remember the epidermal cells are ciliated, so they're kind of wafted away. You're continually making this stuff. It's forced into the third, down to the fourth, and out in the subarachnoid space, all around the cord, all around the brain, and it's absorbed by the arachnoid villi into the dural sinuses. That's the circulation of the cerebrospinal fluid. Okay? At any given time, there's about 200 millilitres of cerebrospinal fluid floating around the system. Any given time, if I have to drain all the cerebrospinal fluid out, I have about 200 millilitres of it. It's about 5-6 fluid ounces. So you've got about 200 within the system, but you make 500 a day. So you make double that. At any given time, you make twice as much as you've got per day. You're making about 500 millilitres of this stuff a day by these choroid plexuses. So there's a continual build-up of pressure within the ventricles. That's the pressure pumps it out and squirts it around the system. It's absorbed as fast as it's made, obviously, in the arachnoid villi. If you do not get it absorbed as fast as it's made, what's the problem? Or, if you get a blockage in the median aperture or cerebral aqueduct, what's the clinical problem? Hydrocephalus, right? Water on the brain. It's not water, it's cerebrospinal fluid. Hydrocephalus is when you get a build-up of cerebrospinal fluid pressure within the brain. The ventricles swell. Either you're making too much, you're not absorbing enough, or more often you're getting blocked somewhere. No big deal for us particularly, well it is, but for children, their bones the skull bones aren't fused. So there's a little child who's got hydrocephalus. The fontanelles just give and the bones expand as the pressure from the ventricles force the brain tissue outwards. And there's another kid who's got hydrocephalus. Various things to do, but typically they'll put a shunt in, right? You might have seen kids with shunts. They basically stick a pipe, a plastic pipe with valves on it, into one of the ventricles and bleed it out. Not bleed it out, but you siphon that cerebrospinal fluid out of the ventricles and down into the peritoneum, down into the stomach, usually somewhere around here. There's various shunts you can put in. These are a couple of examples of them, where they'll take it out of the fourth ventricle here, out of the lateral ventricle here, and they pump it into the stomach somewhere, where it's just reabsorbed. Okay? And that is outside the skull. As this picture shows here, there's a bloke with a shunt. They've got his lateral ventricle here, and there's a pipe basically stuck around the outside of his skull into his neck, and it's going to drip cerebrospinal fluid out of his head constantly to relieve that pressure. I'm sure you'll probably, many of you when you go out in the clinic, you'll see these. You'll see kids. What kids typically have this issue? What clinical diagnosis? Spina bifida. Kids with spina bifida often will have concurrent hydrocephalus, and therefore some of them will have a shunt put in. Here's a little tip, clinically. If a kid comes in and parents say he's misbehaving today, you're treating this kid who's got spina bifida, and he's normally happy-go-lucky kid, whatever, and the parents come in and say he's feeling bad today, he's irritable, treatment won't be very good today because he's in a bad mood, or he's sick, or they'll say he's a bit dizzy today, he's confused today, or I don't know why, his balance is off. So slow symptoms start developing. First thing you think, his shunt is blocked. These valves get blocked. A little valve in his pipes that stop backflow of cerebrospinal fluid, and they can get quite easy. It's very common to get a blockage in a shunt. And the first thing you'll see is a kid's behaviour, motor coordination is off that day. And it can get worse and worse and worse. That kid needs to have that pressure relieved, those valves need to be changed, that shunt needs to be addressed by a surgeon quickly. That build-up pressure is going to go on and on and on and on and on. And the behavioural change is going to get worse and worse and worse, and coordination, etc., etc. I once saw an 18-year-old kid, I think I've mentioned this in anatomy, in England, who came in complaining of a headache. There was nothing wrong with this child before. She was 18 years old, this girl, and she had nothing wrong with her apart from a headache, and maybe felt not quite right. CT scan showed this. Here's the head, typically like whatever. There's the bone. This was a ventricle. The whole of the center of her head was one massive ventricle. Her brain had been squashed gradually through 18 years against her skull. And the whole center of the brain was one massive pool of cerebrospinal fluid. So her brain was plastic, and as that pressure built over 18 years, it adapted, and the brain was literally compressed to like half an inch maybe width of brain tissue around her skull. The inside of her skull was one massive ventricle. And all the kid was complaining of was a headache. That just shows how plastic the brain can be. Obviously this built up. This obviously pressure started to build up for some reason after the fontanelles fused. So nobody knew. If it occurred before the fontanelles fused, you'd have seen, you'd have seen a kid like this, and you'd have known, oh, something's going on here, let's fix this. We had no idea this kid had this issue. Amazing. And headache was what she came in and complained of. You saw this CT scan, bloody hell. How are you alive, kind of thing, you know what I'm saying? Sometimes, last thing I said, well, cerebrospinal fluid is often tested for to see what's in it. As I said, if you lose sugar, you often have a bacterial infection. That's a key sign of a bacterial infection, because they need sugar for energy, bacteria. The worst one to get, of course, is streptococci. That's a gram-negative stain bacterial infection. Do you remember my gram-negative, gram-positive cell biology? You talk about gram-negatives and gram-positive stains? No? Well, I won't talk about it. If you see anything, anyway, one thing. Gram-negative, serious bad stuff. If something's a gram-negative stain showing up on their chart, they're in a bad way. Gram-positive, not so bad. Just one fact to remember. If you see a gram-negative stain bacterial infection, they're in a serious mess. Bacterial meningitis. There's been outbreaks of that in England lately. That's a medical emergency. It's often fatal, spinal meningitis. There's been cases in America recently, and I've seen outbreaks of it for some reason. And you basically get a bacterial infection within the cerebrospinal fluid affecting arachnoid layer and pia mater. Serious stuff. You're right within the nervous system now. Symptoms of bacterial meningitis, what would that be? Unfortunately, it sounds a lot like a flu. Neck pain, high fever, headache. That could be spinal meningitis, it could be a flu. Okay? One of the key things you'll see, often will distinguish it from a flu, is this neck guarding. What do you mean by guarding of the neck? They stiffen up, right? Like crazy around the neck. Why do they do that? Because if you did this, it would hurt like crazy, and that was what sign? Limit sign. You get that shooting electrical pain because you're stretching the meninges, right? You're stretching and they're inflamed. You do this, it hurts like crazy. And that's a classic sign of spinal meningitis. That could be, hopefully, it's not, it could be bacterial meningitis, and they're in a very serious mess if you've got that, because they could die quite quickly from that. Okay, let's move on. Looking at the cord itself now. Okay, we've gone through meninges, we're now at the cord itself. Let's look at this one. I like my diagram better. Thought sensor. Okay, here's a cross-section of the cord, a generic cross-section of the cord, okay? On the outside you have white matter. White matter, why is it white? Myelinated neurons, fat. Okay? So the outside, the white stuff around the outside of the cord are tracts going up and down. Those are often called columns. The white columns surround the grey matter inside, the butterfly or the H of grey matter. Okay? So white stuff are myelinated neurons called columns going up and down. So you've got ascending and descending tracts within these columns. Okay? On the back, we have what's known as the dorsal white columns. The dorsal white columns are on the back of the cord. That's the top of the diagram. And they are split in half by the posterior median sulcus. It's off the top of the screen, but I think hopefully you printed that. The posterior median sulcus is the crack in the back of the cord. Okay? And around there we have our dorsal white columns, or posterior columns. These are dorsal. Okay? On the front we have our ventral white columns, or anterior white columns. There's a bigger split there. There's a bigger split in the front of the cord than in the back, and that's the anterior median fissure. A sulcus, as in the brain, is a small ditch, a small crack. A fissure is a big crack. You see a fissure, it's a much deeper. So the anterior median fissure separates these ventral white columns. On the outside, of course, we have lateral white columns. Simple as that. Lateral white columns, dorsal and ventral. Simple. Those, again, are pathways for stuff going up the cord or stuff coming down the cord. Okay? In the center, we have this butterfly H pattern, what are you going to call it? Of grey matter, non-myelinated neurons, cell bodies. These are connections, left and right. Okay, that's where the neurons connect. These are horns. Columns white, horns grey. Dorsal grey horn, ventral grey horn, lateral grey horn. So dorsal at the back, lateral next in the middle, sort of a side, and then ventral at the front. The grey commissure is the cross piece of our H, of our goalpost. The cross piece is a grey commissure. Commissure fibres, what's the biggest commissure fibres in the brain? Connect left and right? How is our left hemisphere connected to our right hemisphere? What's the biggest chunk of fibres connecting left and right? Tough body, corpus callosum. Corpus callosum, remember that? Those are commissure fibres connecting left and right. The grey commissure in the cord connects left and right parts of the cord. Right down the middle, that blue dot I drew, is our central canal, cerebrospinal fluid going down there. Next one picture, there it is, the central canal. Okay, very quickly, remember our sensory fibres come in the back of the cord, our unipolar neurons come in the back of the cord, there's our dorsal root ganglion, they go into the back of the dorsal root. Out the ventral root come our motor neurons. Our motor neurons are alpha and gamma motor neurons. Those cell bodies are going to be found within the ventral grey horn there. The ventral grey horn is our alpha motor neurons cell bodies here. They come out the ventral grey horn and they form our spine, or they form C5, whatever it is. Okay? Let's see what happens. So a few of these columns. What happens? They are named based on the function of these. So this diagram shows that those white columns, within those white columns we have pink and blue bits. Okay? Those pinky bits on the left hand, on the right hand side of the diagram are our ascending tracks. Those are our sensory tracks going up to the brain. They've all got names, they've all got various functions. We'll go over these in a minute briefly. The blue things on the left are our descending tracks. Have you come across this diagram before? You don't know the location of our tracks in the cord? Okay. Let's look at our ascending tracks, some of them. We're going to go over these in detail as we come, when we start the detail part of this course. This is, remember, hopefully a rough overview of your A&P courses. So it's a very cursory introduction to these pathways. And there's another diagram of that. We'll come back to that. Alright. Here is a couple of our ascending tracks. Okay? Each sensation comes into the cord to the dorsal root. They ascend in the cord through one of those columns to get to the brain. The type of sensation it is dictates where within the cord it goes up. So it's random. Okay? So, let's look at the sensory pathways over here for proprioception. On this side, on the left hand side, is the pathway for proprioception. And here's a key point. Whatever sensation it is, pain, temperature, proprioception, stretch, pressure, vibration, whatever it is, it gets through the sensory cortex through 3 neurons, a chain of 3. Whatever it is. There's millions alongside each other, but in sequential, there's 3 of them. And They're known as first order, second order, third order. Okay? And the location of these changes. Okay, let's just briefly look at this and we'll stop. The first order neuron is the neuron that takes the sensation from the skin, the muscle, the tendon, whatever it is, to the cord. Okay? That's the first order neuron. The third order neuron, I'm skipping a second for now, the third order neuron always goes from the thalamus, the contralateral thalamus, to the somatosensory cortex. Do you know where the somatosensory cortex is? Do you know what I mean by the somatosensory cortex? Please say yes. That's a basic. The somatosensory cortex, right? No? It's directly behind the central sulcus. Right? It's directly behind the motor cortex. Remember the strip on the frontal lobe called the motor cortex? Just behind that is our sensory cortex. It's a strip across the brain. And we'll talk about that later when we get to the details of it. But there's that strip across here. Every sensation, apart from our special senses in our brain, you know, our sight, etc., go to there. So my right hand goes to my left somatosensory cortex. My right foot, my right thigh goes to my left somatosensory cortex. And it goes there by a series of 3 neurons. First order neuron into the cord. Third order neuron, thalamus to cortex. Now unfortunately the second order neuron, how it goes, changes as a function of what sensation it is. That's where it gets tricky. Let's just look at one of these and we're going to stop. Proprioception. Proprioception. From this person's foot here. It comes in through the dorsal root. It then takes a upward bend and goes up. It doesn't stop. The axon carries on upwards in the dorsal columns. Goes all the way through the cord, all the way up to the medulla. This is proprioception only. It ascends ipsilaterally. Second order neuron takes over the medulla, crosses, and goes up to the thalamus. What time am I supposed to stop? 5-2 or 10-2? 5-2. Okay, so we'll stop it. First order neuron into the cord. Second order neuron up the cord to the medulla. Third order neuron. Sorry. No, I was wrong. Let's stop. We'll go over that again in detail next class. Meanwhile, read about it and you'll be unconfused.